So let me go ahead and start, and I'm going to try to move fairly quickly, but I'm also going to try to leave some time for questions at the end. I did put in a disclosure slide. And the key points that I want to touch on are just, why it is that we have these immune stimulatory effects. The immune system does things for a reason. And I'm going to make a point that I think is especially important. It's come up a little bit, but that these responses often, but not always, are species specific, and talk a little bit about what those species differences are. Just speak to the point about formulations and modifications, and then finally coming back to screening, and I'll try to add a few other points beyond those that you've already heard. So why did the immune system evolve to be activated by nucleic acids to begin with? The first reason may or may not be pathogen detection. That's obviously one important function that these defense pathways serve. And the fact is that certain foreign nucleic acids have modifications that can be detected by these pathways. The CpG we talked about, especially in the past. In our genomes, when C is followed by G, it's usually methylated. In bacteria and viruses, it's not methylated. And TLR9 evolved to detect that. As has been mentioned by a couple of the speakers, in phosphodiester DNA, this detection is relatively specific for unmethylated CG. In phosphodiester DNA, TLR9 is not really activated by methylated DNA or by other sequences to any great degree. In phosphorothioate DNA, that is no longer true. A methylated CpG will still activate TLR9 in phosphorothioate DNA. Not as efficiently, but it still activates TLR9. TPG activates TLR9 in phosphorothioate. Polypyrimidine sequences activate TLR9 quite well in phosphorothioate DNA. And so that's why you have these episodes of finding oligonucleotides that you get into human clinical trials, and you're seeing immune stimulatory effects that were not detected before. Human TLR9 detects different sequences than mouse TLR9, slightly differently around this. And the cytokine responses are different in humans and mice. Modified RNA can also activate separate pathways, and I'll come to that. Unmodified RNA normally is excluded from endosomes during infection. It's present there, and so that can be a sign of infection. And then the 5' triphosphate or diphosphates on the 5' end of RNA can activate the RIG-I pathway. And so when that occurs, the immune system evolved very rapid defense pathways that evolved to detect against viruses and bacteria, especially viruses. Antiviral defense mechanisms are more preferentially triggered by those pathways. Now, a second important mechanism for these, and it may have actually evolved first, is stress detection in cells. When cells are stressed or when tissue is damaged, nucleic acids escape from their normal compartments. Double-stranded DNA comes out of the nucleus into the cytoplasm. RNA can get into compartments from which it's normally excluded. And so some of these pathways may serve homeostatic mechanisms in forming cells or an organism that a tissue is damaged and leading to responses that could determine, is there a pathogen also present in this damaged tissue? Or is it simply damaged tissue and all we have to do is to repair it? How all of this comes together is not clear, but it may explain why there are so many different pathways here that are evolved for this. And in terms of the tissue damage, I think the STING pathway may be particularly important there, and I want to spend a few minutes to talk about that. Self-RNA or foreign RNA in endosomes can activate these TLRs. I didn't include TLR3 here because usually therapeutic oligonucleotides are short enough that they don't activate TLR3, but with longer RNAs, mRNAs, that could become an issue as well. And these other pathways induce inflammatory responses that are very similar but sometimes different from the responses that you get with the first pathways I talked about, and I'll come back to that. So let me just spend a minute talking about species specific results. This is an overview from a very nice review article by Gunter Hartmann and Martin Schlee of the various DNA sensing immune pathways. And you can see in the upper left in the endosomes is where TLR9 can interact with CpG containing DNA. So TLR9 activation does not require endosomal escape. And that makes sense as a defense mechanism. As soon as DNA is internalized, it can activate TLR9, and within 7 minutes, you can start detecting NF-kappa B activation in B cells, for example, that have taken up CpG DNA. The intracellular pathways, several different pathways converge on STING, and the STING pathway was initially identified at leading to IRF3, that's interferon response factor 3, and to the production of interferon in the nucleus of cells. And then the AIM2 pathway in the cytoplasm leads to inflammasomes. That's a separate pathway that is involved, also can lead to inflammation with IL-1 beta production and pyroptosis. Now, one of the species differences here is that in human cells, especially myeloid cells, this was first reported last year, the STING pathway not only activates IRF3, but it also activates the inflammasome pathway. And so for screening purposes, if you're screening in rodents, you could miss that. In terms of thinking about the efficacy of the STING pathway for various applications, that's also an important thing to know. Rodents may not predict that accurately. In the case of TLR9, we've referred to this before. In mice, TLR9 is expressed in many different immune cells, most notably monocytes and macrophages, whereas in humans, it's expressed only in 2 cell types that have been well documented. If you read the literature, there will be reports that it's expressed in T cells and NK cells. I really don't think those reports are correct. We've never been able to document functional expression in those cell types. So functionally, we think that TLR9 is really only in plasmacytoid dendritic cells. These make interferon, and in B cells. And the B cells can make various inflammatory cytokines as well. The pathways in B cells and PDCs are a little bit different, and the PDCs are fragile cells. So when you're screening for the immune effects, if you use frozen PBMCs, for example, PDCs don't tend to survive that, and so you won't get activation of those cells. B cell lines, you cannot mimic the activation pathway that occurs in the PDCs because that pathway goes through IRF7, which is not present in B cells. If you're using the 293 cells that are transfected with TLR9 as a readout for your assays, those cells use an NF-kappa B reporter that does not detect the activation pathway that occurs in PDCs also. So certain CpGs can activate PDCs without activating B cells. Now with phosphorothioates, this isn't really a problem because the phosphorothioates activate B cell TLR9 so well, and they don't actually activate the PDCs. But if you're developing phosphodiester DNA, then you need to be aware that the PDCs are a cell type that you need to think about as well. In mice, TLR8 doesn't seem to be functionally active. So when you're screening for RNA immune effects, as I mentioned before, TLR3, TLR7, and TLR8 are all TLRs that can be activated by RNA. TLR7 and TLR8 are the major ones that we worry about with screening oligonucleotides because TLR3 typically requires a longer sequence, probably about 30 bases or so in humans. The downstream pathways again lead to interferon production and production of inflammatory cytokines. And so, let me just go back. There's one other point I wanted to make on the previous slide here. So on the right-hand side, I made the comment that in order to detect immune activation through any of the innate immune pathways, you have to think about the downstream outputs that you're going to look at. And to comprehensively detect activation of the inflammasome pathways, the interferon response pathways, and NF-kappa B, which leads to TNF, IL-6, and other cytokines. At a minimum, you need to screen for interferon alpha or beta, or even more sensitively than interferon alpha or beta, are the downstream interferon-inducible chemokines. And CXCL10 is one that's commonly used, also called IP10. It is actually a more sensitive indicator of interferon production than the interferon itself, because the interferons tend to get consumed, especially in vivo. And so in the serum in vivo, you will not see interferon induction from immune stimulatory oligonucleotides at concentrations where you will see induction of IP10 or CXCL10. Secondly, you need to look at the cytokines and chemokines triggered by the NF-kappa B pathway. IL-6, TNF are good sensitive indicators of those. And then the inflammasome pathway, IL-1 beta, is a reasonably good indicator for activation of that pathway. So if you screen for all of those, then you more or less have those covered. So let me just make a few comments about the formulations and their effects. Formulations, of course, are very often used. They can improve the PK, biodistribution, getting cells into other tissues. And there's clearly an interplay between the formulations and the cargoes in terms of the immune effects. And a very nice sort of accidental example of that is the type of lipid nanoparticle that's called Smarticles. And this lipid nanoparticle has been used in the clinic by 3 different companies to deliver different nucleic acid cargoes. Pronai has used it to deliver DNA, and they've reported mild to moderate immune symptoms from their use with this. Myrna is a company that's no longer in existence, unfortunately, and that is because they were trying to deliver a microRNA mimic with this using an unmodified RNA. And the cytokine release syndrome that occurred in humans was so profound that they had to pre-treat patients with high doses of steroids in order to just enable dosing. And they were never able to find a therapeutic index, and the company eventually failed. Mina currently is using this in the clinic now with a modified RNA backbone that does not have nearly the immune activation that you get with an unmodified RNA, and that appears to be generally well tolerated. So although the formulations themselves do have immune effects, the cargo that you put in the formulation is really key. We've talked about the phosphorothioate backbone and its effects, increasing protein binding and immune stimulation. But really, almost any charged backbone appears to have some level of immune effects. The phosphorothioate is just the most studied. What is clear is that uncharged backbones, and the morpholino is the one that is in widest use, really dramatically reduce or even eliminate the immune effects. Morpholinos at very high doses don't seem to have any TLR interaction. You don't see cytokine induction in humans or rodents for that matter. And I believe the same thing is true with the peptide nucleic acids. Sebastian made the point, which I'm really glad that he made the point, that phosphorothioate DNA that does not contain any CpG can still activate TLR9. So I won't dwell on that. And then with the 2' modifications or the bicyclic modifications, those certainly reduce the immune effects of oligonucleotides, but again, they don't completely eliminate them. Now, you don't have to completely modify an oligonucleotide with 2' modifications in order to greatly reduce the effects. But you do have to think hard and screen hard on which sites in the oligonucleotide you make the modification. Because just shifting a modification from one position in an oligo to an adjacent position in the oligo can dramatically change the toxicity and the efficacy profiles. And this is something that I think everyone doing these kinds of experiments has discovered. I don't know of any ways to predict how those effects are going to occur. And so the only way you can really find it is to just make a lot of oligos and screen them. So let me turn now to screening of the compounds and just talk briefly about that. What to screen for, I mentioned before, sort of a short list there, and then an optional longer list. And this I'm starting with in vitro. Basically, we use Luminex, or you can use MDS. I don't think you have to do a whole Luminex 25-plex panel. Doing an MDS assay for 6 or 7 analytes is probably enough. But when you're setting up your particular system and platform, unless it's really identical to other platforms where this is well worked out, you'll want to keep an open mind for what factors are the most sensitive ones to detect immune effects in your system. Human PBMC is our go-to assay. I will add, we use about 10 times the number of cells that most other groups seem to use. And the reason we've done that is because it's more sensitive. I think the reason it's more sensitive is because the PDCs, the plasmacytoid dendritic cells, seem to like to be really crowded. And so if you want to get them the happiest and get the highest interferon induction, you have to use 10 times as many cells as you normally do. And you need to use the round bottom or U-bottom plates. Flat bottom plates, you don't get nearly as sensitive outcomes there. I think I mentioned you can't use frozen PBMCs, or at least not very well, and this is an issue we're trying to get around. I'd love to have a cell line that gave adequate results. In our hands, we've never found a cell line that we were happy with, but not to say it can't be done. And it depends again on what you're doing. And for our purposes, we're looking for immune stimulation, and we're using phosphodiester DNA, and we're very dependent on the PDCs. So if you're doing something different, then a B-cell assay, cell line assay may be perfectly adequate. We've only done a little bit of work on whole blood assays, and I know some groups use those all the time and are very happy with them. In the limited experience we had, whole blood assays were just as sensitive as the PBMC assays once we optimized them, but it required 10 times the amount of DNA, and so we just went back to the PBMC assays. Some people use mouse spleen assays. I don't like mouse cells just because the immune effects are so different from human cells, so we just stick with those. And in terms of the time point, you can start to see cytokines within a few hours in the supernatants, but we found the most sensitivity using a 48-hour collection of the supernatants. 24 hours may be enough, but 48 just seems to be a little more sensitive. In vivo, we do screening in rodents with just injecting 3 or 4 mice per group and collecting serum. Typically, if it's a naked oligonucleotide, we do a serum collection at 3 or 4 hours. 6 hours seems to be fine. But in our hands, with phosphorothioate oligos, by 24 hours, most of the cytokines are back down to baseline. And so, you just have to think about those time points. We have found with formulations, sometimes the timing changes, and then a 24-hour collection is appropriate. I'm just making all of these very detailed points on the assays because so often I've read papers or heard people presenting at meetings where they say, oh, no immune effects. And they've looked at a single time point in an assay which is insensitive, and you just, if your work is being reviewed by Paul, for example, he's going to blast it. So you really want to think about these things. And then for hepatotoxicity, I think we've talked about that a fair amount. We also like roughly 100 milligrams per kilogram over a couple of weeks as a dose to look for that. Hepatotoxicity can result from a lot of different things. Immune stimulation is one. That definitely CpG oligos can cause hepatotoxicity, but obviously other things can too. And then renal tox screen, I think I'll skip that. And I wanted to stop here, and I will answer one question that someone had asked me recently, is what about screening for CNS tox with oligos and CNS immunotox? And actually, I asked Art Levin and Doug Kornbrust, do you have an answer for that? Because, frankly, we haven't done a lot of screening there, and so I just don't have a good answer for CNS screening tox. But with that, if we have time, I'm happy to take a few questions.
Would you measure IP10 in CSF?
I would, absolutely, yeah. Because I think the cells that produce IP10 are in the CSF. Now, I will add, in rodents, glial cells are macrophage and monocyte derived. And rodent glial cells do appear to express TLR9 and respond to CpG. I don't believe that primate or human glial cells do, but I'm not positive about that.
This is a conversation that started about 9 o'clock Friday night. It's timely. Go ahead, please.
You can shout and I'll repeat the question. Okay. Yeah, immune suppression of oligonucleotides. So there are like poly-G oligos in particular can have a variety of suppressive effects. And there are some oligonucleotides that can be antagonists for TLR9. That may be what you're referring to. Those are not really well understood, actually. And we were very interested in trying to develop an immune suppressive oligo clinically for treating autoimmune disease or other conditions. And I don't know if it's a problem of the pharmacokinetics or what, but we did not get consistently positive enough results that we felt we could take that into human clinical trials.
Yeah, I was asking that because we've been looking at two primal metal modifications, and basically they sequence-specific patterns that inhibit TLR7, TLR8, TLR9, and even TLR3. And obviously, I'm presenting this work on my poster, but I think it's really something people don't appreciate. I guess it's easy to see something that goes up, so when you look at immunostimulation, but immunosuppression is highly important. I mean, we all know of like the anti-TNF drugs, which can promote TB relapse. So it's like something you don't see because you don't think about it, but that actually could be very bad. People that get the flu, if they have no TLR7, for instance, they'll get maybe a lethal infection. So.
That's a good point. I'll look for your poster. I think people normally are just looking for immune stimulation and not thinking about that, but interesting point. Thanks.